let's resume from where we left from the first part of this lecture on photosynthesis. We talked about the light reaction, how through the creation of this electron flow, thanks to the help of light, basically leads to the formation of lots of ATP molecules by chemiosmosis through ATP synthase and storing some energy in NADPH. Now we're going to move to the next step. How is this energy used to fix carbon dioxide into glycerol halide 3-phosphate? So what we talk about now is the Calvin cycle here. Quick overview of the Calvin cycle is basically a series of reactions that basically lead to the fixation of carbon dioxide into glyceraldehyde 3-phosphate. So carbon dioxide is a 1-carbon molecule. Glyceraldehyde 3-phosphate is a 3-carbon molecule. So you need 3 of these cycles happening. So 3 of this carbon dioxide molecule in order to produce 1 molecule glyceraldehyde 3-phosphate. Another thing I want to stress here is, look, there is no glucose. We oftentimes teach photosynthesis as the reverse of cellular respiration, but it's incorrect. The end point of photosynthesis is not the creation of glucose, but is the glyceraldehyde 3-phosphate, which then later on can give origin to glucose through reaction external to photosynthesis. But it can give origin to other monosaccharides. Okay, so it's not glucose the end point of all of this. This is the molecule we're talking about here, glyceraldehyde 3-phosphate, which this slide is taken from glycolysis. If you recall, is the same molecule that is produced as an intermediate in glycolysis after the first 5 energy requiring reaction. So that's the molecule produced by photosynthesis. And then that's the one that later on in the second part of glycolysis, in the energy releasing part, gives origin to pyruvate. Now you can see from this slide here too, how producing glyceraldehyde 3-phosphate in photosynthesis then later on can be used to produce glucose outside of photosynthesis again, but other monosaccharides such as fructose. So if you start from here, you get fructose 6-phosphate out of this, then you can actually produce fructose. You can produce other molecules. You can produce glycerol. That's the entering point of glycerol from fatty acid, from triglycerides. And so glyceraldehyde 3-phosphate is the end point of photosynthesis and photosynthesis. It can be used to generate many other molecules other than glucose. Okay. So let's focus on the Calvin cycle. Now we have all this ATP and NADPH from the light reaction. And so in this Calvin cycle, it's used through 3 different steps to fix basically carbon dioxide into glyceraldehyde 3-phosphate through these 3 steps: the fixation, reduction, and regeneration. So the first step, fixation, that's where actually carbon dioxide gets fixed to another molecule, which is ribulose 1,5-bisphosphate, which is regenerated by the cycle. And every 3 cycles, you have 1 molecule that gets regenerated here. And this molecule here, so this fixation of carbon dioxide to ribulose 1,5-bisphosphate is done by this enzyme called Rubisco. Memorize this. We'll talk about that later on. We'll see that there is a problem with this enzyme too. So this carbon dioxide gets fixed to ribulose 1,5-bisphosphate, forming 2 molecules of 3-phosphoglycerate here. So that's referred to as the fixation. Now, then this molecule, 3-phosphoglycerate, gets converted ultimately into glyceraldehyde 3-phosphate through the next step, which is referred to as a reduction. Series of reactions there too, where the phosphoglycerate gets first converted into 1,3-bisphosphoglycerate, and then it produces glyceraldehyde 3-phosphate. Now, notice one thing. You don't need to remember all of these steps, but you need to remember the different phases and what they do ultimately, what's the point of them. But what I wanted to show you here is this is where most of the ATP and all of the NADPH produced by the light reaction get used. You have 6 ATP molecules used here for every 3 cycles and 6 NADPH molecules used for 3 cycles. We see the stoichiometry later on to generate glyceraldehyde 3-phosphate. Once reduction is done, now it doesn't produce only glyceraldehyde 3-phosphate, and that's all of that enters. Some of the glyceraldehyde 3-phosphate actually stays into the cycle because in order to fix more carbon dioxide, you have to produce more ribulose 1,5-bisphosphate. And so hence the last part of the cycle called regeneration, where some of the glyceraldehyde 3-phosphate, actually most of it, if you look at, is actually converted into ribulose 1,5-bisphosphate. And in this case too, requires some energy in the form of ATP to be added here, and which is coming as well from the light reaction. So in the 3 steps, we have the fixation, that's where carbon dioxide gets fixed to ribulose 1,5-bisphosphate. Then you have the reduction, which leads to the formation of glyceraldehyde 3-phosphate. And then the last step, which is regeneration, where ribulose 1,5-bisphosphate gets produced again so that the cycle can keep going through that. And through all of this, the ATP and the NADH produced by the light reaction get used to sustain this cycle here. What about the stoichiometry? This summarizes it in words. It's easier to see it in a pictures. If you look, now we know that the endpoint is 1 molecule of glyceraldehyde 3-phosphate. This is a 3-carbon molecule. So what it means is that in order to produce one of these molecules, this cycle has to fix 3 molecules of carbon dioxide. So it has to take place 3 times. Okay, so you need 3 of these cycles and 3 carbon dioxide molecules in order to produce 1 molecule of glyceraldehyde 3-phosphate, which then also produce 5 additional molecule of glyceraldehyde 3-phosphate, which then are used to produce 3 molecule of ribulose 1,5-bisphosphate. So altogether here, you need 3 cycle to produce 1 glyceraldehyde 3-phosphate. And because of this, each cycle we use 2 ATP here and 1 ATP here. You need altogether 9 ATP molecules to produce 1 glyceraldehyde 3-phosphate, 6 here, 3 here, and 2 NADPH per cycle, so 6 NADPH molecule to produce 1 G3P molecule, glyceraldehyde 3-phosphate molecules. That's the overall stoichiometry of this process. Now, again, the endpoint of the Calvin cycle is glyceraldehyde 3-phosphate, which can then later on be used to produce several monosaccharides, not only glucose, but also fructose, other molecules there, which then can be used to produce disaccharides, to produce polysaccharides such as starch and cellulose, and so on. So it's just the endpoint is glyceraldehyde 3-phosphate that can be used to produce many other things outside of photosynthesis. Last thing I'm going to discuss here is one problem that there is with photosynthesis. And the problem is with this enzyme, the first enzyme of the Calvin cycle, which is Rubisco. You can learn the full name here if you want to look smart. However, you can just remember the name Rubisco for this enzyme. That's all I need you to remember here. This is the enzyme that is responsible to fix carbon dioxide to ribulose 1,5-bisphosphate in the first phase, which is fixation of the Calvin cycle. It's an extremely abundant enzyme, which is responsible for about 50% of all the proteins in the leaves, produce a lot of carbon dioxide, it removes a lot of carbon dioxide, blah, 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 all these things. Now, these enzymes, structurally speaking, so we practice a little bit of what we learn about enzymes, is actually made by 16 subunits, with 8 identical one referred to as small subunits and 8 identical one referred to as large subunits. Now, the active site is in the large subunits. Okay, this is where the active site that binds to carbon and ribulose 1,5-bisphosphate, this and this is bound by the large subunit. Large subunit that is actually synthesized by the chloroplast genome. Remember that small genome that is present inside the chloroplast is actually synthesizing this unit here. The other unit instead is synthesized from the chromosome in the nucleus. So it's the small subunit is there, and it doesn't have the active site, but it has important regulatory effect on the large subunits. Now, what's the problem of this enzyme? The problem with this enzyme is that it's an old enzyme. It's an old and extremely important enzyme, which basically means when it comes to proteins, they're very old but extremely important. The effect of evolution in them, they tend to evolve much less than proteins that don't have a huge importance. So this enzyme has been there doing photosynthesis for billions of years. Okay. And it's so essential, is the key entry point, that it cannot mutate too much, right? And one of the consequences is that being so old, it has evolved in an environment which is very different than the environment that we have right now. More specifically is the relationship with oxygen. This enzyme has evolved 3 billion years ago with the first photosynthetic organisms in an atmosphere that basically had no oxygen, right? Very rich in carbon dioxide, but no oxygen. So it could easily bind to carbon dioxide and do its job. Now, with time, the amount of oxygen in the atmosphere has grown more and more and more and more, thanks God, so we can survive. What is the problem with that? Is that Rubisco has not evolved in the meantime. And even without knowing, between brackets there, it actually had high affinity for oxygen. So with time has not changed, but the presence of increasing presence of oxygen creates a problem because now it has another molecule it can bind to very efficiently. And the problem is that when it binds to carbon dioxide, it does its job. It does the first part, the first phase of photosynthesis. It does the fixation part. Beautiful. But then if it binds to oxygen instead, which now is very abundant, it doesn't produce any more 3-phosphoglycerate, but instead it produces phosphoglycolate too. Produce both phosphoglycerate but also phosphoglycolate, which then in the plants get converted into glycolate, which is very toxic for the plant. And so plants have to actually break down glycolate into carbon dioxide again through a process referred to as photorespiration, which requires the use of energy. So basically what it means is that nowadays this enzyme is not fit anymore to perform the Calvin cycle efficiently because it loses a lot of energy by producing a product that then has to be broken down through photorespiration, right? So plants and other photosynthetic organisms like algae had to develop, since they could not, evolution did not allow to change Rubisco too much because it's an essential enzyme. Any mutation that lead to the ruining the function will lead to disappearance of species. So now they have evolved different strategies to overcome this problem. And so just sorry to frame it again here. So the affinity of Rubisco for carbon dioxide is still much higher than oxygen. So it's still a good enzyme in that sense. However, the problem is that the oxygen has become so much more now in the air compared to carbon dioxide, 440 times more, that there is a problem now. And so that's the problem, that this leads to Rubisco binding oxygen more than carbon dioxide, and therefore to photorespiration, energy loss, and so on. So now, no matter what organism you're talking about, algae or plants, now they have evolved some strategies to overcome this, which ultimately what they play with is the relative amount of oxygen versus carbon dioxide. If this is, let's assume, the current ratio, 540 times, at least in air, in water is slightly different, of oxygen versus carbon dioxide. Now, how they overcome this problem is by enriching the environment in which Rubisco acts of carbon dioxide. And they've done it with different strategies. So by doing that, now you have Rubisco exposed to a lot more carbon dioxide than oxygen, then tendential will buy carbon dioxide and do its job during the proper Calvin cycle. How have they done that? Well, algae, they found a very simple solution overall. Solution that involves only basically two proteins, a pump and an enzyme. So now, even in water, there's a problem. There is a lot of oxygen that is present compared to carbon dioxide. So if algae just relied on the simple diffusion of oxygen and carbon dioxide from the outside to the inside, they would end up having too much oxygen compared to carbon dioxide inside. So how did they enrich the inside in carbon dioxide compared to the outside? Well, they do that using a pump that actually pumps bicarbonate from the outside into the inside. This doesn't affect the movement of carbon dioxide. It's a different molecule. So bicarbonate gets pumped inside, and as soon as it gets inside, now there is an enzyme called carbonic anhydrase that converts bicarbonate into carbon dioxide. So by doing that, now you have the normal diffusion of carbon dioxide, but on top of that, you have more carbon dioxide produced inside by bringing in the bicarbonate. So at this point, the ratio between oxygen and carbon dioxide inside, near to the chloroplast, where Rubisco is, has been altered so much more in favor of carbon dioxide, so that Rubisco would preferentially bind carbon dioxide than oxygen, and therefore photorespiration will be minimal, if not completely absent. Plants have evolved a different strategy. Now, plants have also other problems other than compared to the algae. So they had to evolve a different strategy compared to algae. One of the main problems that they have is that together with photorespiration, so due to the abundance of oxygen, they have to deal also with water loss. They're not in water, right? So they are exposed to air. So water tends to come out from the plants. How did they deal with that? We'll see also in BIO359, they dealt with that by coating basically their leaf with wax. So producing what is called a waxy cuticle, a layer of wax, which is hydrophobic and therefore restricts the movement of water from the inside to the outside and vice versa. However, this obviously reduces the movement of water, but reduces the movement of gas as well. And so they had to come out with a second strategy to allow the movement of these gases and actually control it as well. And this is done by having stomata. Stomata are opening on the leaves. Usually they're on the bottom part of the leaves. You don't see them with your naked eye, but you can see them under a microscope. These are made by two cells actually, called gate cells, which they can swell or they can shrink. And by doing that, they open and close these opening, this stomata. Okay. So when they're open, they allow for the gases and some water to come in and out. But when they close, they prevent the loss of water and oxygen and so on, and the gases. So by having these stomata now, these plants have the time to control the water loss. They have a way to control the water loss and at the same time to exchange gases. However, they also limit the amount of time in which they can take the carbon dioxide in, which is necessary for the Calvin cycle. And together with that, they get oxygen in. So how do they limit photorespiration within the limit of time in which these stomata are open? Well, they've done that by developing another cycle, which is referred to as the C4 cycle. This C4 cycle is a cycle, you see later on the steps, where carbon dioxide, instead of being brought into the Calvin cycle directly, gets first fixed to a 3-carbon molecule, forming a 4-carbon molecule. By doing that, they have a way to accumulate as much carbon dioxide as possible. I'll show you in a second. So to understand it clearly, this is the situation. Imagine if this is a plant that doesn't have the C4 cycle, this C4 cycle that binds to carbon dioxide. So what would happen here is that when the stomata, so these openings are open, now you would have that the carbon dioxide from outside moves in, all right, and it will move in by diffusion until you have the same concentration inside than the one you have outside. The same thing would happen for oxygen. So you would end up having a relative amount of oxygen and carbon dioxide inside, similar to the one you have outside, right? Obviously, this is a problem because these will lead to Rubisco binding mostly oxygen and therefore also performing, not mostly, but a lot of oxygen, performing a lot of photorespiration, a lot of energy loss. Now, with this cycle, C4 cycle, which I show you what it is in the next slide. Now, some plants that do this cycle too, they have a way also to bind this carbon dioxide as soon as it comes in. So let's say this is open for a few minutes. When this carbon dioxide coming in, immediately gets bound through the C4 cycle. By doing that, basically they keep this inner concentration of carbon dioxide low, which means this flow keeps going, keeps going, keeps going, right? So for the limited period of time in which the stomata are open so that there is no water loss, they can bring in a lot more carbon dioxide than they would if they didn't have the C4 cycle. And what happens then once the stomata get closed, now through this cycle still, they can actually release this carbon dioxide back. And now they can perform the carving cycle with much more carbon dioxide compared to the oxygen that's come in. So they have, at this point, changed the ratio between carbon dioxide and oxygen inside compared to the one on the outside, therefore favoring the binding of rubisco to carbon dioxide and minimizing photorespiration. Now, these plants that do the C4, not all plants do the C4 cycle, okay? And so those that do it are referred to as C4 plants, and those that don't are referred to as C3 plants. Now, not surprisingly, lots of these C4 plants are plants that are they found themselves in very hot and dry climates, or very hot climates as well. So climates that do favor the water loss. All right. So climates that basically forces them to keep their stomata open for a very short period of time. Together with that, though, other plants in temperate climates as well, such as corn, sugarcane, have developed that too, because by doing that, they actually can become much more efficient overall at fixing carbon dioxide and therefore to do the calving cycle. Okay, so by doing this, by adopting the C4 cycle, these plants, whether they're tropical or dry plants, they actually have maximized the amount of carbon dioxide they can take in and became more efficient and do this calving cycle. Now, here is the C4 pathway that I talked about. What does it consist of? So this pathway is nothing else than a series of reactions, like in the calving cycle, but this is not about permanently fixating carbon dioxide. It's not about producing glycerol halide 3-phosphate. It's about temporarily fixing carbon dioxide so that you can release it later on. And so how do they work? Well, carbon dioxide, imagine this is the situation where the plant has the stomata open. Now there is carbon dioxide coming in. Carbon dioxide through the C4 pathway in these C4 plants is immediately fixed to PEP, which stands for phosphoenol pyruvate. And this molecule then leads to the formation of a 4-carbon molecule called oxaloacetate, C4, 4-carbon, hence the name C4 pathway. So by doing that, basically this cycle removes the carbon dioxide from the air. And so more carbon dioxide can come in through the stomata. This keeps going until the stomata are open, and now they close. The stomata close. What happens next? So now you have, so this molecule has been converted into malate first, which accumulates inside the leaf. And then when the stomata are closed, now malate can actually be reconverted into a 3-carbon molecule, in this case pyruvate. By doing that, you have release of that carbon dioxide that originally came in and was fixed through the first step. So by doing that now, with stomata open, you remove as much carbon dioxide as possible. With stomata closed, you can re-release this carbon dioxide. So then now you can alter the relative concentration of carbon dioxide versus oxygen, and then you can perform the calving cycle in a much more efficient way with rubisco binding carbon dioxide instead of oxygen. Now, depending on how the plants do that, or basically the location of these two cycles, the C4 cycle is the one we just mentioned. The C3 cycle will be the calving cycle. This can be performed within the same cell or in different cells. So some plants, they do perform this cycle in 2 different cell types. Look at this. This is a section or a schematic of a section of a leaf. So this will be the epidermis of the leaf. These are the stomata. That's where the air comes in. So some plants do have cells here referred to as mesophyll cell or mesophils that, not surprisingly, they're around this opening. where the stomata are. And so these cells here are the cells that do perform the C4 cycle. So as soon as some carbon dioxide comes in, not surprising, they fix it, right? Producing malate here. And then once these stomata are closed, now this malate is transferred to the other cells here around the vascular bundles here. These cells are referred to as the bundle sheath cells. And these are the cells that instead do perform the Calvin cycle. So now the malate releases the carbon dioxide here, forming pyruvate, C3 carbon, three carbon molecule. Carbon dioxide is released here, and now Rubisco has a lot of carbon dioxide to bind to and perform the Calvin cycle in the bundle sheath cell. This produces glyceraldehyde 3-phosphate, which then can be released in the vascular system here and brought to different part of the cells, or can be converted into glucose, other monosaccharides here, and then released into the vasculature of the plant. So in this case, since there are two different types of cells that do the C4 cycle and the Calvin cycle, you talk about spatial separation of the C4 pathway and the Calvin cycle. Some other plants, which are dry plants, such as this one here, they're called crassulacean plants, have developed instead a different type of separation, which is referred to as temporal separation. So the C4 pathway and the Calvin cycle in these plants is actually done in the same cell type, but it's done at different time. That's why it's called temporal separation. These plants are referred to as the CAM plants or crassulacean acid metabolism plants because it was first discovered in these type of dry plants called crassulacean plants. So in this case, so when the stomata are open, so not surprising, these plants are exposed to very dry and hot environments, right? So they want to minimize water loss. So they don't open their stomata very frequently. And they open them only basically at night when the air is cooler and it's a little bit more less dry. Okay, so they open them at nighttime for a short period of time, and so these cells, when at night the stomata are open, they do the C4 cycle. They fix as much carbon dioxide as possible into malate, and then as soon as the sun or the light comes out, they close the stomata. Air is getting too hot and dry, so the water will come out. You close them. You don't want to lose water, and then they revert from the C4 cycle. The malate instead releases this carbon dioxide, which is then is used by Rubisco to do the Calvin cycle. So they do the Calvin cycle during the day. So at different time, they do the same cells. They do the C4 cycle at night, C3 cycle or Calvin cycle during the day. And therefore, we talk about this as temporal separation. So we're done with this lecture, putting together the first part and the second half. So what did we discuss? We first went through an overview of photosynthesis. We talked about the organisms that do that, the photoautotrophs. Then we talked about the organelles, where they do it, the chloroplast, their structure. We talked about these key components, the pigments, how do they absorb light, at what wavelengths, and why you have different types of them. And when we talk about the photosystem, how they are organized and what is their function, which ultimately is to make the light reaction exergonic from endergonic. Then we talk about the light reaction. We put everything together and we discuss how the electron flow in the light reaction is made, first of all, exergonic from endergonic by absorbing 2 photons from each electrons that come from water. It gets transferred to NADP+, producing NADPH. All of this is done with the goal of producing a protomotive force, which, like in oxidative phosphorylation, is then used to produce ATP by chemiosmosis using ATP synthase. And then this leads basically ultimately to the formation of ATP and NADPH, which then can be used in the Calvin cycle to fix carbon dioxide into glyceraldehyde 3-phosphate, process that is done through 3 steps: fixation, reduction, and regeneration. Finally, we finish up talking about the problem with the first enzyme of the Calvin cycle, which is Rubisco, which is involved in the fixation step, problem that comes from his high affinity for oxygen, something that ultimately leads to or deviates the activity of this enzyme away from a proper Calvin cycle, deviates it towards ultimately photorespiration and energy loss. How is this overcome? We talked about how algae overcome it and how particularly how plants overcome it by generating a different cycle, which is referred to as the C4 cycle, which can be done by different cell type in spatial separation or by the same cell type in temporal separation of the C3 and the Calvin cycle.